You should get excited because we're going to talk about science. I'm very interested in biomedical engineering, and I'd like to pursue a degree in this field. While pursuing this degree, I'd like to study organ transplants, in particular the synthesis of human organs for transplant. According to the U.S. Department of Health and Human Services, approximately 121,000 people were on an organ donation waiting list in 2013, but only 29,000 transplants, 24%, were performed with organs given by 14,000 living or deceased donors. We're suffering from a shortage of available organs. On average, 22 people die each day waiting for an organ transplant. This dilemma has led doctors to turn to xenotransplantation to solve the organ shortage. Xenotransplantation is when non-human organs are transferred into a human body. Pig organs are commonly used in research and would be the best to use in a human. Think about that the next time you eat bacon. Xenotransplantation doesn't use organs from other primates because there's too high a risk of transferring a novel virus into the human population, since primates like chimpanzees are too genetically similar to humans. Pigs are not genetically similar enough to humans, so that risk is much lower. There is one caveat. Porcine endogenous retrovirus is present in every pig, and the retrovirus can infect human cells in vitro, or outside the body. The retrovirus, however, has not infected non-human primates in research. Researchers are unsure if porcine endogenous retroviruses are pathogenic, and more research needs to be conducted on these retroviruses before xenotransplantation can have widespread use. The genetic difference between pigs and humans also means that recipients of porcine xenotransplants need to take immunosuppressants so their body doesn't reject the organ taken from a pig. Immunosuppression leaves people susceptible to a whole host of germs in their environments. People who receive human organs also need to use immunosuppressants because the genetic code of the donor is not exactly the same as the code of the recipient. This and the risk of porcine endogenous retroviruses are why researchers are working on genetically modifying pigs so organs with human DNA can be grown in swine. This would mean that there would be a lower chance of rejection from the recipient's body, especially if the organs are grown with the recipient's own DNA. If we are readily able to grow organs in swine, or even 3D print organs, the organ shortage would be mitigated. I would like to be part of the initiative to make whole organ xenotransplantation a reality and help save lives. But first, I need an education.